Afternoon student, today we will discuss some of the important point in translation, particularly translation regulation. So first point will be upstream open reading frame and how it's regulate translation. Then how the suppressor tRNA it function. Then one new concept, what is a chimeric RNA named as tmRNA. Now we will start with upstream open reading frame and how it regulates translation. To understand that thing, thing I want to mention, like it's an eukaryotic polypeptide chain synthesis when it's happening. Don't think that always it is starting from its first AUG. Sometimes the first AUG is not present according to the frame. In other way, I can say that what is written here, in other words, some upstream open reading frames, which is actually named as small u, represent upstream ORF open reading frames, are present before the main open reading frame. Okay, so what actually wanted to tell you, not all eukaryotic polypeptides are encoded by an open reading frame that start with AUG that is most proximal to the 5' terminus. In some case, the first AUG is not in a proper sequence context, resulting in the bypass. That means, so you just see this, this diagram, then you will understand. The green colored horizontal line is an mRNA. What they wanted to tell, that this is the AUG. This is the AUG which we are talking, and this is the stop codon. This is the mRNA which 5' is capped. Now this is the main ORF, but sometimes before the main ORF there is an upstream small ORF also may be present. And because of that, what may happen? The ideal condition is that, the normal or ideal condition is that translation will initiate with the first AUG, that is the upstream open reading frame, and when it encounters the stop codon, it will automatically stop. So it starts in normal case, and then once it reaches UGA, which is a stop codon, it should dissociate. But in rare case, this smaller subunit of ribosome, instead of getting rid of this first upstream open reading frame, but it still attached and start scanning and then reach to the first AUG of the main open reading frame, and thereby doing the translation. Continuously translation is going on. So the third diagram, if you see, the first one is actually in the normal case what it should happen, but in this one, the rare one, what we are showing that instead of dissociating from the mRNA, this small subunit which is still bound with the initiator tRNA started scanning and will reach to the main AUG sequence. If this thing is happening, then always a leaky translation is going on. Okay, when the first one is going on, at the same time there's a little bit of translation is always on because of this reason. So this is how the upstream open reading frame occupy sometimes the smaller subunit of ribosome with the initiated tRNA, and because of that, the main translation open reading frame, always a translation is happening. Here comes another scenario. There is the second one. This one is generally happen for viral mRNA when it start producing its protein. So instead of having a real ribosome entry point, it carries what a sequence named as IRES, internal ribosome entry sequence. There's somewhat similarity with the prokaryotic sequence, which, because of this sequence which is present, this entire pre-initiation complex can come and bind to this internal ribosome entry sequence. If you remember eukaryotic translation initiation, all this 40S, 48S will come and bind to the cap, and with the cap it will start entering there. But here unfortunately what is happening, as there is an IRES sequence is there, this will recognize this internal ribosome entry sequence and will start the translation. So what we are showing here, we are showing an AUG. After that there's a stop codon, but this is the main our frame which we are talking. But before that there is an internal ribosome entry sequence, IRES, where with the help of, if you remember, eukaryotic initiation factor 4F is a 3 subunits factors A, G, B. These proteins are present here with this F, and then with all the other initiated tRNA, other GTP binding factors, it comes and binds with the 40S complex and this internal sequence, and because of that translation initiate. This situation generally takes place for viral infection when viruses need its mRNA to be synthesized, translated more efficiently. Okay, so this is how the internal sequence guide the ribosomal subunit to initiate translation with more efficiency. The previous one shows the leaky translation. The next one is showed that bypassing everything because of not following the actual rule, it recognizes an internal ribosome entry sequence. This is an hairpin loop-like structure. The internal ribosome entry sequence is a hairpin loop-like structure. It mimics the cap. So it starts its translation from the internal side. Now after that we will discuss the suppressor mutation can reside in the same or different gene, and suppressor tRNA, how these things are happening. Now we will start with something that if there is a mutation, there is an alteration of the gene function, you will see the alteration of the gene function. But sometimes what happens, if another mutation takes place in the same gene, okay, it may revert its function back. Okay, so some of these subsequent mutation are easy to understand, being simple reverse or back mutations which change an altered nucleotide sequence back to its original arrangement. That's what I say, that if there is an alteration, we say there is a mutation, and if somehow that mutation is rectified, we will say it's back mutation. In some case, you can think like there's an alteration in, there's a missense. You understand what is called missense mutation? There's a wrong amino acids inserted. Because of that, the complete three-dimensional structure of the protein impaired, and that's not functional. But if another mutation somehow restore a partial arrangement, 3D arrangement of the protein, and we sometimes may get back the function, that's also we can consider the reverse or back mutation. This type of things you can see we call suppressor mutation, and it falls into two main categories. One is called intragenic, that means within same gene. Another one intergenic. Intragenic meaning one point mutation happened, because of that is non-functional, and the second mutation happened in the same gene but other side. Okay, because of the second mutation you may get back the protein function again. That we are telling intragenic. Another one is that intergenic, that means mutation takes place in one gene, but another mutation in some other gene may restore the first gene's function back. That we call intergenic. With this knowledge, let us discuss some of the suppressor mutation category. This is we call a suppression of frame shift mutation. In this diagram, you will see a deletion in the nucleotide coding sequence can result in an incomplete inactive polypeptide change. So this is a DNA sequence, a gene, a gray color, and there is a deletion, the bluish part showing a deletion of a nucleotide during gene replication. So we are getting mutant DNA 1 they are mentioning, and because of that this is we are getting a mutant mRNA 1, and because of this a missense codon, because frame shift has happened, because there is a deletion, so some wrong amino acids are coming, that's we call missense codon, and then there's a nonsense codon. Because of that, some, if we are considering the protein, some correct amino acid definitely will be there at the 5' side, then some, because of the missense codon, some incorrect amino acid, and then truncated. This is incomplete and abortive chain ending, so this is a faulty protein we are getting. Now how the suppression can take place? This is intragenic within the same one. Now if we consider this mutant D1, this mutant D1, we are here, we are generating mutant D1 because of a deletion mutation, correct? Now come here, mutant D1 we are taking. Now again another mutation takes place. Now this time it's an insertion of a nucleotide in gene during gene replication takes place. So in a same gene, two mutations we are talking. The first one is the deletion, the second one is the insertion. So because of this insertion, now we are telling it is mutant DNA 2. What kind of mRNA we are getting? Definitely a mutant mRNA 2. First part green, then definitely a missense amino acids are coming. Then because of this insertion, then again the frame came back. Open reading frame came in the correct order. So some faulty amino acids are there, but after that it restored. Now because of that, this small change, maybe this protein gets back its function, its three-dimensional structure is correct. So this mutation, because of this deletion, there is a mutation and we are getting a faulty protein, but a second mutation in the same gene restore its active site and we are getting active protein. So this is called a suppression. Now this is given an example with intra. Now we will discuss something inter, meaning some other case, other genes mutation, how it affects it. Now see, this is this will call nonsense suppression. What is that? This figure shows how a minor tyrosine tRNA species act to suppress the nonsense codon in mRNA. So nonsense meaning the stop codon. So as we know, there is no tRNA present which can recognize a nonsense tRNA. But imagine if some equally it is present. We all know that some tRNA, genes are there. Because of some mutation, this tRNA now able to recognize the nonsense codon, that means the stop codon, with an amino acid. Now what will happen in that case? Now see this left hand side, what we are talking. This is a mutated gene containing a nonsense codon. So this is DNA 5', 3', 3', 5'. See correctly. So because of that we are getting an mRNA. So this is DNA, that's why TAG. If it comes as a RNA, T will be read by UAG. So this is UAG is a stop codon. So because this is a stop codon generating, because of a mutation here we are getting one UAG mRNA. So there will be no protein against amino acid against that. But imagine, it happened in E. coli in some cell, there is a mutation in gene coding for minor tyrosine tRNA. So this actually a gene for a minor tyrosine tRNA. Generally tyrosine tRNA are specific, but there are some alteration takes place in some type of tRNA that instead of in the anticodon sequence. See, this is a gene coding for minor tyrosine tRNA. Tyrosine tRNA recognizes the tyrosine codon 5' UAG 3' and 5' UAU 3'. So this is should be UAU. Instead of that, it will recognize now UAG. Now it is a tRNA I'm showing, so 3', 5'. Remember, this is 5', 3'. So somehow, because of the mutation, now this tRNA can recognize this UAG, which is a stop codon. So this is a UAG stop codon, which is coming from the mutated gene because nonsense mutation takes place here. But here what happened? A minor tyrosine tRNA, which is carrying the tyrosine amino acid, instead of recognizing UAU, now it is recognizing UAG. See, it is always coming in a reverse manner. So this is 5 prime 3 prime. If RNA is 5 prime 3 prime, tRNA will be opposite 3 prime 5 prime. So you are seeing AUC. So it can recognize a nonsense codon, and it is coding for whom? Tyrosine. So it is inserting a tyrosine residue where it should not insert anything, but it is inserting a tyrosine residue where our codon is UAG. So the nonsense codon is suppressed. Tyrosine is inserted at the position of the nonsense codon, that is allow the formation of a complete polypeptide chain. So because of this mutation, there is a termination of protein synthesis. But instead of stopping the protein synthesis, what I am seeing here, there is a minor tyrosine tRNA is there who can recognize the stop codon and inserting tyrosine. So that's called intergenic, because it's a two different gene, and because of this mutation, this is get suppressed. Understood? So suppressor tRNA, this is a suppressor tRNA because it is suppressing whom? A nonsense codon by inserting a tyrosine residue. Now the third one is another type of regulation where the situation is what? Translation dependent regulation of mRNA and protein stability. The situation is something here. An mRNA is translating inside the ribosome, but the mRNA is truncated at the 3 prime terminus. Nothing is there. So from the beginning it is started as usual normal translation with the help of ribosome and all other factors. But what happened at the end? There is no stop codon. So if there is no stop codon, there is no releasing factor will come and bind. Ribosome cannot be released. mRNA is trapped. All the tRNA, they are trapped inside. But it is a situation where all these things are together, but it cannot proceed. Why it cannot proceed? Because there is no stop codon. See the diagram, you can see this green color is the mRNA, this is the ribosome, where it is continuous protein synthesis going on. As this mRNA is truncated, that means 3 prime side, there is nothing is there, there is an empty. So this cannot be rescued. To overcome this problem, in bacteria, there is a situation. If this situation arises, then only bacteria synthesize this type of chimeric RNA. Why chimeric? Because this type of RNA, they are having a part of the RNA mimicking a tRNA, and other part is having kind of an mRNA. So that also they named as tmRNA. Let me read little bit. At this point meaning if this is the situation arises, the ribosome cannot proceed. There is no codon either to bind an amino acid tRNA or to bind a release factor. That's what I was telling also. Without some mechanism to release them from the defective mRNA, many ribosomes would be permanently trapped, removing them from polypeptide synthesis in prokaryotic cells. Such stalled ribosomes are rescued by the action of a chimeric RNA molecule that is part tRNA and part mRNA. So that's the named as tmRNA. Remember, who is coming to rescue this situation? This is called a chimeric RNA. They named as tmRNA because partly it is resemble tRNA, and other part it has mRNA like similarity. So a classical example is that SSRA is a 457 nucleotide tmRNA that includes a region at its 3 prime end strongly resembles tRNA alanine, and it has it can bring the EF2 GTP and can form a SSRA alanine EF2 GTP and can binds to the A site of the ribosome. And after that, what happened? it transfer the growing polypeptide chain. If you see from this place, is enough room to for these big chimeric RNA to enter through its which is called the alanine, resembling alanine tRNA, okay, and carrying an alanine amino acid also. Now there will be a true peptide bond synthesis. The growing polypeptide chain now will be transferred on alanine. So this tRNA will get released. Now what will happen? This chain will be here. Now the tmRNA, the example of that one tmRNA is SSRA, which is a 457 nucleotide long. So it is now carrying the polypeptide chain. Now how it will end? It has a stop codon. If you see this, one part resembling tRNA, another part resembling what? mRNA. Now protein synthesis will carry on based on this new mRNA part of this chimeric RNA, and there is a stop codon. With that, it will end the protein synthesis, and what will happen? At least 10 amino acid tag, they mentioned this a tag, will be incorporated in the growing polypeptide chain, and this is the red color. That's why they showed these amino acids are faulty. That's not part of this the regular one. So these amino acids, as that's why they mentioned tag. Because of this tag, some protease will recognize and destroy the protein, destroy this entire protein. Okay, but at the same time, ribosome will get released, and the mRNA which was trapped get released. All the tRNA other also get released. So you need to think like that. If this is the situation, first you need to know which situation this kind of chimeric RNA will come. If there is a truncated mRNA and translation already started, then to rescue this thing, chimeric RNA, which is named as tmRNA, because partly tRNA and partly mRNA, and it will first do the peptide bond synthesis, that transfer this thing, transpeptidation reaction it will do, and it will take the help of, if you remember, the elongation factor GTP to GDP, and after that the regular protein synthesis will be going on with the help of this mRNA portion of the chimeric RNA, and when it encounters a stop codon, it will stop the translation normal mechanism. Okay, but the protein which will getting N terminal, maybe the correct amino acid should be there, and the C terminal some tagged 10 amino acid will come, and that can be recognized by a protease and will degrade the entire system. So only when the situation is something like that, then only the E. coli will synthesize this kind of chimeric RNA and rescue the situation. Okay, with this today's lecture I'm ending. Hope you understood. So any problem, difficulty, can give me a call.